Hi students, I am Dr. Mashur. Welcome to my class. Today I would like to discuss bacterial DNA replication. Okay, bacterial DNA replication. DNA replication. DNA replication is the process of copying of genetic material that is known as DNA. Okay, DNA replication is the process of copying of genetic material DNA. Okay, and there is a bidirectional DNA replication is occurred in both prokaryotes and eukaryotes. Okay, there is bidirectional replication is occurred in both eukaryotes and prokaryotes. Prokaryotic chromosomal DNA. Okay, bidirectional means this replication will be occur in both direction of DNA, and many enzymes and proteins that are involved in DNA replication. Okay, in both eukaryotes and prokaryotes, there are many enzymes and proteins that are involved in DNA replication. Okay, there are many enzymes and proteins that are involved in DNA replication, and we have discussed about these major enzymes and proteins which are involved in DNA replication in our previous class, and replication of the bacterial chromosome. Okay, replication in the bacterial chromosome that is tightly coupled with the bacterial growth cycle. Okay, the bacterial DNA replication, bacterial chromosomal replication is tightly coupled with the growth cycle of bacteria, and the synthesis of DNA molecule is a complex process involving series of steps. Okay, this DNA replication is a complex process and that's involved series of steps. Okay, in both prokaryotes and eukaryote DNA, chromosomal DNA replication having mainly three steps. Okay, both eukaryotic and prokaryotic DNA replication having mainly three steps. One is initiation, second step is an elongation, and third step is a termination. First step is initiation, second step is an elongation, and third step is a termination. Okay, DNA replication is a process of copying of genetic material DNA, and the bidirectional DNA replication is occurred both in prokaryotes and eukaryotic chromosomal DNA, and many enzymes and proteins are involved in DNA replication, and replication of bacterial chromosome is tightly coupled with the bacterial growth cycle, and the synthesis of DNA molecule is a complex process and it is involved in series of steps, and both eukaryotic and eukaryotic chromosome replication have mainly three steps. First one is initiation, second one is elongation, and third one is termination. So we can see these steps separately in detail. The first step, initiation. Initiation means the starting of DNA replication, and initiation of replication occurs at the site called origin of replication or ori. Okay, origin of this initiation is occurred at the site called origin of replication or is called as ori, and this origin of replication having the ability to replicate autonomously. Okay, this origin of replication having the ability to replicate autonomously, and DNA molecules that replicate from an origin of replication that is called as a replicon. Okay, a DNA molecule that's replicate from an origin of replication that is known as a replicon, and this origin of replication have high content of adenine and thymine. Okay, this origin of replication is an AT-rich region. Okay, this origin of replication have contain high amount of high concentration of adenine and thymine nucleotides, and this prokaryotic chromosomal DNA having only one origin of replication. Okay, this prokaryotic DNA only having one origin of replication. That's why it's called as a unireplicon. Okay, this prokaryotic origin of prokaryotic chromosomal DNA contain only one origin of replication. That's why it's called as a unireplicon. And but in the case of eukaryotes, there are thousands of origin of replication is present, because compared to prokaryote, the eukaryotic DNA is much large compared to prokaryote. This eukaryotic DNA is much large in size. That's why there is thousands of replication, origin of replication is present in the eukaryote. That's why it's or eukaryotic chromosomal DNA is called as a multi-replicon. And E. coli chromosomal origin of replication RIC contain 4 9 base pair sites for the initiated protein DNA. Okay, this origin of replication of E. coli chromosome contain site for binding of initiated protein DNA. Okay, initiation of replication is occurred at a site called origin of replication, and this origin of replication having an ability to replicate autonomously, and the DNA molecule that originates from the origin of replication, that is called as a replicon, and this origin of replication is an anti-rich region. And in the case of prokaryote, there is only one origin of site, replication site is that's why this is called as a unireplicon. And an eukaryotic case of there is thousands of origin of replication is there, and each chromosome contain many origin of replication. That's why it is called as a multi-replicon. And the E. coli chromosome contain a site for binding. The E. coli chromosomal origin of replication contain a site for binding of protein, initial protein DNA. In the initiation process, there is a protein called DNA. Okay, initial protein DNA that with ATP bind over the origin of replication of bacterial chromosome. Okay, this is DNA protein with the ATP that bind over the origin of replication of negatively supercoiled DNA. Okay, that's DNA protein with the ATP that bind over the origin of replication of negatively supercoiled DNA. Okay, this negative supercoiling of DNA is achieved by the enzyme called DNA kinase that will remove the positive supercoiling of DNA and it will convert to negatively supercoiled DNA. Okay, this DNA protein bind with the origin of replication of negatively supercoiled DNA, and it will facilitate the melting of sequence percent. Okay, it will facilitate the melting of sequence and which open the double stranded DNA. Okay, which open the double stranded DNA. Here you can see that's DNA protein that bind or the negatively supercoiled DNA. Okay, negatively supercoiling is occurred by the action of enzyme DNA kinase. Okay, that will remove the positive supercoiling from the DNA and that will convert to negatively supercoiled DNA. Okay, the binding of this DNA A protein on the basal DNA that will leads to the melting of double stranded DNA. Okay, you can see in this figure the double stranded is melted and that is opened the double stranded DNA and that forms a special structure that is called replication problem. Okay, and this melting that will leads to the formation of a special structure that is called replication problem. Okay, replication problem is formed here by the melting that is caused by protein DNA A. And this opened DNA onto this open DNA, another protein that is called DNA B, and is called as DNA helicase. DNA B or is called as helicase. DNA helicase that will bind on this open DNA. Okay, that will bind on the DNA helicase will bind on the open DNA here, and here you can this DNA helicase will be bind. Okay, and this binding of DNA helicase we already defined that DNA helicase the activity was removing double strand the hydrogen bond of double strand DNA, hydrogen bond of double strand DNA. This DNA helicase will remove the hydrogen bond of double strand DNA that will leads the expansion of this bubble and it is form a specialized structure called replication fork. Okay, replication fork, and this DNA replication is bidirectional in chromosome. That's why here you can see one replication fork. Here also one replication fork because this is a bidirectional one one replication will be gone to this direction and the one will be gone to this direction. Okay, replication fork is a specialized structure that form during the process of replication. One DNA guided is some DNA helix remove the double hydrogen bond between this two DNA strand, and it has a two branching prongs. Like a fork, it has having a two branching prongs, and each strand will act as a template for synthesizing new DNA strand. Okay, it will act as a act as a template for synthesizing new DNA strands. Okay, and this replication fork. Okay, this replication fork we have given here. Okay, we have given here. Okay, and we have given here, and we told that DNA helicase bind that will formation replication fork. Okay, you can see here different enzyme. One enzyme is the DNA kinase, which removes the supercoiling, supercoiling, and give the negative supercoiling to DNA. And here one enzyme is the helicase enzyme that will remove the hydrogen bond. And when the binding of DNA helicase, that will lead to the formation of two single strand DNA. Okay, two single strand DNA here you can see, and this two single strand DNA on this two single strand DNA. Okay, there is one protein is called SSP protein. One protein is called SSP protein. Okay, you can see one protein is called SSP protein. That is SSP protein. Is a single strand binding protein will bind on this single strand, and that will protect the single strand from the cleavage, and it also prevent the reunion of these two strands. Okay, it will prevent the reunion of strand, and this SSP will protect the DNA from the cleavage. And here you can see one primer, RNA primer. This RNA primer is synthesized by DNA primerase enzyme. Okay, DNA primerase enzyme. And here only one RNA primer will be there. And here you can see different many DNA RNA primer will be there. You can see here. This also synthesized by different RNA primer that also synthesized by DNA primerase enzyme. And this primer is for the synthesis of leading strand, and this primer is synthesis of lagging strand. Okay, this is about the initiation of DNA replication. And the elongation process. In the elongation process, this helicase enzyme continue to unwind of DNA like a zip open. Okay, this helicase enzyme will move this direction, and top of this guy is also move in this direction in the front of helicase, and this helicase will remove the double strand this hydrogen bond and is unwind the DNA like a zip open. Okay, this helicase will remove the hydrogen bond and is open the double stranded DNA like a zip open. And during the process of elongation, the DNA polymerase 3, DNA polymerase 3, that will synthesize DNA daughter strand on the 5' to 3' direction. Okay, DNA polymerase 3 will synthesize the daughter strand in 5' to 3' direction. Okay, here you can see the 5' to 3' direction will synthesize. Okay, 5' to 3' direction will be synthesized, and one strand will be synthesized towards the replication fork. Okay, one strand will be synthesized towards the replication fork. Okay, one strand will be synthesized towards the replication fork, and that strand, this strand will be synthesized towards the replication fork, and this strand is known as a leading strand. Okay, this strand is known as a leading strand. Okay, and here you can see one different primers is the RNA primer is there, and this strand will be synthesized away from the replication fork. this strand will be synthesized towards the replication fork, and this strand is synthesized towards the replication fork, and this strand will be synthesized away from the replication fork, and from this prime. From this primer, this strand is synthesized for a small piece, and here also one strand is synthesized, and here also synthesized. Okay, this strand is known as lagging strand, because lagging strand is not a continuous synthesized strand, and this leading strand is a continuous, but is a semi-discontinuous. So this is a discontinuous strand. Okay, this discontinuous strand is known as a lagging strand. Okay, this discontinuous strand is a lagging strand, and this lagging strand contains many fragment of DNA. That fragment is called as a Okazaki fragment. Okay, this small DNA fragment is called as a Okazaki fragment. Okay, and this DNA fragment is called as a Okazaki fragment. Okay, sometime in exam they will ask what is Okazaki fragment. Okay, this small DNA fragment is contained present in a lagging strand that is known as an Okazaki fragment. Okay. And this Okazaki fragment having 1000 to 2000 nucleotide size. Okay, this Okazaki fragment having 1000 to 2000 nucleotide size. Okay, this Okazaki fragment is present in the lagging strand of DNA synthesis, and lagging strand is synthesized away from the replication fork. Okay, here you can see this leading strand is synthesized in continuous way, and this lagging strand is synthesized in discontinuous way. That's why DNA replication is semi-discontinuous. Okay, that's why one is continuous and another is discontinuous. That's why DNA replication is semi-discontinuous. Okay, when this DNA, this leading strand and lagging strand synthesis is off, that under one protein enzyme that is called DNA polymerase 1. Okay. DNA polymerase will come, and that will remove the RNA primer present here. Okay. This DNA polymerase 1 will remove the RNA primer present here, and RNA primer present here, and that will fill the gap. Okay. That will fill the gap of RNA primer. Okay. That will fill the gap of RNA primer, and after that DNA ligase will come, and that will seal the nick between these fragments. Okay, seal the nick between these fragments, and after that one enzyme called DNA polymerase 2 will come, and that will proofread the DNA strand. Okay, after synthesis of DNA, that is DNA polymerase 2 will come, and that will synthesize DNA polymerase 2 will proofread the DNA strands. Okay. Is about the elimination. Okay, for exam that time they may ask you this illustrate the replication fork. Okay, illustrate the replication fork. That you draw this picture and you explain what are the enzymes, what are the structures, what are the proteins present here. Sometimes they may ask you in the examination illustrate the replication fork. Okay. Now we can see after elongation, we can see the termination. Okay, in termination occurs in bacteria by two ways. Okay, in bacteria, the replication process occurs by two ways. One is replication is terminated by when the two replication fork is meet. Okay, when the two replication fork is meet. Okay, we are told that one replication fork will move this direction, and another one replication fork will be move this direction, and both will meet in one direction, at one point. Okay, one replication. Okay, here you can see one replication fork will be move this, and another one is moved, and here one point that will meet. Okay, the replication is terminated whenever two replication fork is met. Okay, the replication is terminated. One mechanism is one replication replication is terminated whenever two replication fork is met. Okay, and the other mechanism is the specific termination mechanism is present in bacteria, and one specific mechanism present in bacteria, that is there is a specific termination sequence block the further replication of bacteria. Okay, there is a specific sequence that will block the termination, that will block the replication of DNA in bacteria. Okay, the opposite side of origin of replication. Okay, this coiled DNA, the original this opposite side of this origin of replication. There is a one sequence is called the TER sequence. TER sequence is there. One replication sequence is called TER sequence is there, and on that one other one protein called TES protein will bind. Okay, TES protein that will bind on the TER sequence. Okay, the TES protein bind on the TER sequence, and this TER sequence will stop the replication. Okay, this TES protein that will stop the replication. Okay, how is stop the replication means it's stop the movement of helicase enzyme. Okay, this TES enzyme, TES protein will stop the movement of helicase enzyme. That why this DNA replication is terminated. Okay. This is about DNA replication. I hope you
You understood this class.
thank you for listening.